Hi Gabe! I thought I'd show you what I've been working on these past few weeks for my final project. So this project, it was an idea from my boss. It's something he's been trying to work on for a while and it just hasn't quite worked out. So these two images, they're the same slide in the same spot. It's an image of heart tissue, but you can view it on two different channels on the confocal microscope with two different stains. So this one is a stain for macrophages and this one is a stain for fibroblasts. And what he wants to know, out of these macrophages, which of them are close to fibroblasts and which are not, and in those two groups, what is the morphology of the macrophages in each group, and are they different between groups? So how I did that is I used OpenCV in Python, and the first thing I did was run my code. And I thresholded the images so I could differentiate background from cells, and so I thresholded them till I got to a point that I thought, all right, this is mostly the cells that I want, but you can still see those tiny little background dots that are not cells that I want to get rid of. So I did that with the simple blob detector parameters, and one of the parameters you can do to tell the simple blob detector not to count these cells or to count them is by minimum area of the blob. So I said if it's 100 pixels or less, gone. And then using those parameters, I could detect blobs for the macrophages. I also did this for the fibroblasts. And then you can output an image that draws circles around the macrophages and fibroblasts, so you can see, all right, this is actually detecting the cells that I want. So like you can see, we got some of the bigger cells, but these tinier dots I didn't want to include, and it only counted the ones that I wanted. And once I had that, my data structure, it was pretty simple. And what I wanted to do, when I started, I was thinking in terms of C++ because I have not used Python in a while, is I wanted a list full of structures that were a cell struct, and in that structure would contain all the information about every single cell. And I realized in Python you can't quite do that. So instead what I did is I created a dictionary with just a number for every single cell, and it contains a list with all the information that I want. And the thing with this simple blob detector is that once you get these key points, you can't actually output that much information about them. So here I got the X and Y coordinates. I can get that diameter of the blob, and that's about it that I can export from the key point itself. And then I made some placeholders for where I'll put, is it close to fibroblasts or not, and the average inertia of the cell, which I'll get into in a little bit. And then, so the first thing I wanted to do was figure out which macrophages are within a certain distance of fibroblasts. So I had this dictionary that had all my fibroblasts, just like the X and Y coordinates. It's the coordinates of the central point of the cell. And to do this, to figure out the distance between fibroblasts and macrophages, is I just use the distance between 2 points formula, and I just said if it's less than 100, then they're close to each other, and if the distance is greater than 100, then they are not close to each other. But this is something in the future I'd like to change and adjust to figure out exactly what distance we need, and you have to be able to change it depending on the size of your image anyways. So after I determined which macrophages were close to fibroblasts or not, I wanted to figure out the inertia of the macrophage, and the inertia is basically how circular or elongated the cells are. So an inertia of 0 would mean you have basically a straight line, and an inertia of 1 would mean you have a perfect circle. So the thing with simple blob detector is that even though you can filter by inertia, you can't export that data from the key point. So my workaround for that was to run through this blob detector 100 times, and each time slowly increase the range, like look at a different chunk of the range between 0 and 1 and see what blobs are within that range. So each time I increased by 0.01, and then if the blob was within that range of 0 to 0.01, 0.01 to 0.02, all the way to 1, then I would say it's that the higher end of that range is its inertia. So it's not the exact inertia, but that's the best way I could approximate it using this. So once I had my inertia values and I knew whether the macrophages were close to the fibroblast or not, I turned this into a data frame, just into a CSV if I need to access it later. And then I wanted to create a graph just to see if the two groups were different. So I made a little bar plot, and from this I couldn't see really any difference between the macrophages that were near fibroblast or far from fibroblast. But there are some problems with this code, like the fact that I calculated distance between cells just as the distance between their midpoints, when really I should have had an edge detector and looked at the shortest distance between the two edges to detect distance between cells. I just could not get that to work. So I thought this was a good start, at least. Also, this distance, I don't actually have the size of that image. I don't know whether it was taken on 10x or 20x or what, because these are from a few years ago from someone who's not in the lab anymore, and that would really make a difference too. But anyways, I wanted to take this data and try and test it to see, how accurate this might be. So we have a program that we use called Cell Profiler, and I put in my macrophage image into Cell Profiler, and that can also get you the eccentricity, which is another term for how circular or elongated the cell is. And with this Cell Profiler image, the thing is it got the profile of every single dot, it didn't filter by size. So I had to figure out a way to find which cells are the ones I actually care about and which are just background noise. So I did that I took the CSV that I made in Python, and then I took the 3 columns that I cared about in Cell Profiler, and what I tried to do was see, all right, is there, out of my Python X and Y coordinates, is there anything that seems similar in Cell Profiler that I can assume that's probably the same cell? So, 72, 535, 73, 535, these are probably talking about the same cell. And then 126, 632, 126, 633, these are probably talking about the same cell. And I just went through this whole list in Cell Profiler to try and find the cells that I'm actually interested in learning more about. So once I collected those from Cell Profiler, I had to sort them by whether or not they were close to a fibroblast. So I had those 2 groups, and one issue I did notice is that the way that Cell Profiler calculates eccentricity is that 0 is a full circle and 1 is a line, but the way that Python in OpenCV calculates the roundness of a circle, they called it the inertia ratio, but it's the same thing. They did it the opposite way. So 0 is a line and 1 is a circle. So I just did 1 minus the inertia to get my estimate of eccentricity for my Python data. So I got the means of these 4 groups, whether they were close or not to a fibroblast, in Python and in Cell Profiler, and I graphed them, and they didn't look that different either way, which is a good sign that even though these means are slightly different, these look pretty much the same. So I can assume that the data that I've collected is somewhat accurate compared to Cell Profiler. And the other thing I did is I just put the eccentricity of all of the cells of both groups next to each other just so I could see, how accurate or similar and different they look from each other. And, some of them do not look very similar at all, but some of them look fairly close. So I think I've got something going. I don't think this is, data I could submit for publishing anything yet, but I think it did a decent job exploring what image analysis I can do in Python, and I think I have a good basis for continuing this program and making it better. And the great thing about this is this took me 20 seconds to load, but if I were to use Cell Profiler, each image takes about 10 minutes, and then if I were to use Imaris as another program to detect the distance, which cells are close and which are not, that could be another 10 minutes per image. And here I can just do it all in one place. It's just way faster. I'm just hoping I can get this working, and I think it would be really cool. Do some cool research. And that's all I got, and I've had a good semester. Thanks so much.